Black rot, downy mildew, and powdery mildew are the three fungal and oomycetes diseases that impact grape yield and quality the most in the United States. In the Great Lakes and southeastern growing regions, downy mildew is considered the most destructive of the three diseases. In the western United States, powdery mildew is the most destructive. Black rot, which we will cover in this lecture, is a pesky disease across the United States because of the wide range of temperatures at which the fungus can produce inoculum. It is also the most commonly seen disease in home plantings of grape. The learning outcomes for this lecture are as follows. 1. Know the causative agent of black rot and the signs and symptoms of the disease. Describe the disease cycle of black rot, and finally understand the environmental conditions and timing of ascospore and conidia release. Black rot of grape is caused by the Ascomycetes Guignardia bidwelli. Infections can occur on all green tissue types, although leaf, shoot, tendril, and cane infections rarely result in appreciable damage to the vines. In contrast, berry infections can cause yield losses up to 100% and reduce the quality of the berry. Flower infections result in shriveled flowers. Lesions on the fruit, as shown in the top right image here, are circular with a purplish brown color, almost resembling a bruise. As the lesions expand, they cover the entire berry and then dry up to form mummies, which are shown below. On leaves, irregular shaped necrotic lesions form. Pycnidia are produced within the lesion, which is how you can distinguish the disease from abiotic damage. The pycnidia can be seen with the naked eye. Black rot is a polycyclic disease. The fungus produces parathecia and pycnidia on mummified berries that are on the ground or in the canopy. Ascospores are considered the principal form of primary inoculum, although conidia can also act as primary inoculum. Ascospores are released with the rain and then dispersed by wind and water. Ascospores initiate the disease cycle and cause primary leaf and blossom infections. Infections depend on the length of the leaf wetness period and air temperature during that period. The secondary infections are caused by conidia during the whole growing season, but only if the environmental conditions are favorable. I just mentioned that ascospores are released with the rain and then dispersed by water and the wind. However, the timing of the release of ascospores has been shown to be dependent on the location of the mummies in the vineyard. So the release differs between mummies that are in the canopy and mummies that are on the ground. Ascospores capture studies done in the early 1990s here at OSU revealed that ascospores produced from mummies on the ground are released beginning in early May and continue to be released until the end of June, which corresponds to the end of bloom, as shown here with the yellow line. It's these ascospores that serve as the primary inoculum. In contrast, ascospores produced from mummies in the canopy begin to be released pre-bloom and release peaks at the end of July, beginning of August, as shown with the purple line in this graph. Conidia, which typically function as secondary inoculum, also can serve as primary inoculum when released from overwintered mummies and cane lesions. The number of pycnidia produced and the number that release spores is dependent on percent relative humidity. In the study shown here, after 13 days at 25 degrees Celsius and 100% relative humidity, there was over 300 pycnidia produced per square centimeter on grape leaves, as you can see in this first panel here of graph A. Once the relative humidity goes below 95%, the production of the conidia stops, as you can see here. And I believe I said the production of conidia, but I meant the production of pycnidia. At 100% relative humidity, the pycnidia release the conidia in a slimy matrix called a cirrus, or pluralis cirri. Pycnidia can release several cirri which have a tendril spiral-like shape to them. So again, if you look at the bottom here in graph B, you can see that 100% humidity at 25 degrees Celsius, you get optimal release of cirri, which is that tendril matrix that contains the conidia. Once you get to 95% humidity, you basically have no release of conidia. In this graph, you can see that pycnidia were produced between 5 and 35 degrees Celsius at 100% humidity, but optimal production of pycnidia occurred between 20 and 30 degrees Celsius. So right here, 20, 25, and 30 degrees, you get optimal production of pycnidia, which corresponds to 59 to 77 degrees Fahrenheit. Likewise, in the bottom graph here, graph B, you can see that the pycnidia released the most cirri in this same temperature range of 68 to 86 degrees Fahrenheit. In this short video, the process of a pycnidium releasing several cirri is shown. Notice how many conidia are released from the pycnidium in just 30 seconds. The video is a little over 2 minutes long, however, I'm going to stop it early since you can start to see the release of the cirri and the release of pycnidia before the end of the video.
Grignardia bidwellii, more commonly known as black rot, is a devastating fungal pathogen that affects grapes across the globe. It has the capabilities to destroy an entire harvest if not treated. Black rot initially starts as a small brown spot on leaves, stems, vines, and grapes, as shown here. The disease spreads, causing the brown spot to become larger, eventually affecting the whole grape. The disease will then spread through the vines, infecting the entire cluster, and the tissue turns black and becomes mummified. Sac-like structures called pycnidia form on affected tissue and when exposed to water release spores. Here we have a single pycnidium shown at 100x magnification. Water is added to start sporulation. Spores are immediately dispersed from the surface of the pycnidium. After a brief incubation of less than 1 minute, you will notice a consistent surge of spores exiting the pycnidium in a tendril. After a few minutes, the collective cascade of spores can be seen without a microscope. The amazing observation is that this process is in real time. From our experiences, this is one of the most dynamic macroscopic processes we have ever observed from a microorganism.
Okay, I also posted that video in Carmen, so you can watch the entire video if you would like at a later time. So in summary, black rock can reduce yield by up to 100%, especially when relative humidity levels are above 95% for 24 or more hours. Both ascospores and conidia can serve as primary inoculum, although conidia typically function as secondary inoculum. The timing of the release of ascospores, which serve as the primary principal inoculum, depends on the location of overwintering mummies. Lastly, pycnidia are produced and copious numbers of conidia are released over a wide range of temperatures when relative humidity levels are above 95%. When we discuss the management of fungal and oomycete diseases in a future lecture, these 4 aspects of the black rot disease cycle will be reinforced. Up next, we will be looking at downy mildew of grape.